that we are engaged in the crop improvement. And when you talk the crop improvement, the crop breeding together with other various disciplines, including crop physiology, pathology, entomology, biotechnology. So all of them, they work together, and we have been developing the better varieties with higher resistance to the diseases, higher resilience to the environmental stresses, with enhanced crop productivity, with enhanced pest resistance. And nowadays, nutrition is very key component. So we are working nowadays to enhance the nutrition elements as well. And while doing this thing, we need to ensure the environmental sustainability. So traditionally, breeders have been doing a good job, but until recently, we did not have much advances in the crop productivity. But during last 6-7 years, with the consolidated efforts of the scientists, I think in many crops, many pulse crops, scientists have been able to enhance the crop productivity. But when you are talking about the 32 million metric ton by 2030, it's really important that we need to enhance this crop productivity further. Next, please. And in this regard, and I will be talking something about the modern science, and this is a little bit technical, so we'll not go in the details, but I think that nowadays, and as Robin mentioned and other colleagues mentioned, when we are talking about the climate change, we need to have the varieties which are more tolerant to drought, heat, etc. Now, how to develop them? For that, we can use these modern genomics innovations. How? First, we need to identify the genes which confer tolerance to heat and drought and other stresses. And for that, we can use the genome sequencing approach. In our opinion, we need to have the reference genome for any crop if we would like to implement or integrate these new innovations. Good news is, for majority of the pulses crops, we already got the reference genomes. In some pulses crop, we still don't have, but this is coming up very soon. Now, the next thing is that scientists, either through gene bank or through the breeding program, they got the variety of germplasm collections. So what we need to do now, that we need to use the germplasm on one hand for sequencing and genotyping, so that we can understand, we can go to the gene level. In parallel, we need to phenotype them for those traits in which we are interested, like drought and heat. And then we combine these things together and we can identify genes and haplotypes. Next.